So let's get started. It's my great pleasure to welcome everybody to the UMass iCons program's 8th annual Senior Exposition and Celebration. My name is Scott Auerbach. I'm a chemistry professor here at UMass and the Mahoney family sponsored executive director of the UMass iCons program. Now, I'd love to say that we've never had a senior expo quite like this one before. But again, just like we did last year, we're staying safe and we're going to do this with Zoom. We nonetheless have a really exciting program, and that's a testament to a great group of seniors. The 8th cohort of the iCons program, the so-called 8th color. That's a name that reflects their diversity of research interests, as you'll see, and also they are the largest graduating cohort in our history. Now, we have many special guests to recognize, but at this point, I'm honored to recognize Dr. Tricia Serio, Professor of Biochemistry and Molecular Biology and Dean of the UMass College of Natural Sciences, to give our official UMass welcome. Dr. Serio.
Thank you so much, Professor Auerbach. It's really my honor and privilege to be with you this evening to recognize and to celebrate your accomplishments. Often, our end of the year ceremonies really focus on achievements that can be quantified in terms of courses taken and grades earned. And this viewpoint, to me, really emphasizes the knowledge that you've gained. And while it's certainly important, I think it doesn't really do justice to the time that you've spent at UMass. And specifically, I think the unique ways in which you've pursued that knowledge are just as important as the knowledge that you've gained. To me, the creativity of science, what problems spark our curiosities and how we choose to find answers, often take a back seat to the public appreciation of the rigor and analysis and technical ability most often associated with science. But to me, it's really the creativity that drives science to make a difference in the world. You need to look no further than the COVID-19 pandemic to see examples of scientists from varied disciplines who took their expertise and applied it to tackle this global catastrophe. And in choosing to apply to the iCons program and pursuing this curriculum on top of your major, you've really demonstrated an appreciation for the creativity of science. You've prioritized transforming your education into an opportunity for impact through real-world problems and interdisciplinary teamwork. And you've done so, each one of you, in a unique way. And as you go out into the world, I hope that you remember the lesson that you learned through your participation in iCons. And that is that the world has much to benefit from the ways in which you uniquely choose to use the knowledge that you've gained here at UMass. I especially want to thank the Mahoney family for creating this vision and for their ongoing dedication to making it a reality, and to the UMass iCons team for building such an amazing program with such a strong impact for our students. And most importantly, on behalf of the faculty, staff, and my fellow deans in the College of Natural Sciences, I congratulate all the seniors and all the iCons students on your inspiring accomplishments. It's always amazing end of the year ceremony to see all the wonderful things you've done. You make us proud. Go UMass.
All right, thank you so much, Dean Serio. Thank you very, very much. Wonderful. So in addition to Dean Serio, we have many other special guests from off campus, and these have been strong supporters of iCons. I'd like to thank for coming today. So I'm not sure if he's here yet, but I was hoping that we would be able to welcome Dick Mahoney, a UMass grad. He's the former CEO of Monsanto, and he's really the prime mover of the iCons program back in 2008, before even I got involved. Later on, I know during Senior Celebration that Dick's brother, Bob Mahoney, also a UMass alum, is going to join us and share some comments. Now, we have some folks from the iCons Industry Consortium. This is a group of companies that really get iCons, value the way that we teach, value our students, and most importantly, would like to hire our students. So from the consortium, we have Dr. Peg Crowley-Nowick. She's a UMass grad, is president of Zypher Medical Affairs, and also the chairperson of the advisory board for the iCons program. Also, we have from Waters Corporation, we have Dr. Mike Fogwell and Dr. Nikunj Tana. They are principal scientists at Waters. Waters is also a member of the industry consortium, and Dr. Fogwell is a member of the advisory board. Other members from the advisory board joining us today are Jennifer Fonstad. She's a founding partner of Owl Capital. Ms. Fonstad was named the Venture Capitalist of the Year in 2016, and she's got interests in technology and healthcare. And we have Dr. Ed Merrim, a double alum from UMass in fields of chemistry and physical chemistry, which is my field. And he is presently a business professor at Babson College and formerly president, founder, and CEO of GeoCenters. Finally, we have David O'Connor, who is the former Commissioner of Energy for the Commonwealth of Massachusetts from 1995 to 2007, during the tenure of 5 different governors. He's presently Senior Vice President for Energy and Clean Tech at ML Strategies. So thank you to all of our distinguished guests for coming. I'd like to give them a round of applause. Later on, we're going to have Professor Bob Langer from MIT, who's going to dip in later on. You may know that he is, he's famous for a lot of different things, but he's the inventor of the field of bioengineering. He's the inspiration for what we do in iCons. He's the founder. He started 40 different companies, including Moderna. So you're definitely going to want to stick around to hear what he has to say. So I'd just like to send a warm welcome to all iCons alumni who are with us now, including Erica Light. She's a member of the iCons third cohort, the third axis from 2016, and we're going to be hearing from her later on. Now, we have some dignitaries from on campus as well. So in addition to Dean Serio, who just gave us our welcoming remarks, we have Deputy Chancellor Steve Goodwin. So thank you for coming. He was actually the Dean of the College of Natural Sciences for the first 7 years of iCons. So thank you, Steve, for the successful launch of iCons, for supporting us during that time. And I have to recognize my iCons boss, Associate Dean Mark Tuominen. Got to recognize the boss. Thank you, Mark, for coming today. So we're delighted to have all of you here, along with our iCons seniors, their family and friends, other iCons students and alums, of course, our iCons faculty and staff, all of the research advisors who are here, and all the other friends of the iCons program. So thank you for coming. Now, I would like to echo Dean Serio's comments about the importance of the work that our seniors have done. We are all here to celebrate an idea, and also a very special group of students who've embraced this idea like no others, and in a time like no others. So the idea is simple, really. The question is, what would happen if we gathered together a diverse group of open-minded students and asked them this question: what problems do you want to solve? What's important to you? And how can we all learn to work together, discovering and applying our human diversity to tackle these problems? So we're going to see college students from across the spectrum of majors in science, technology, engineering, and math, STEM, confronting problems in fields, health, biomedicine, energy, environment, national security, and the list goes on. It's an amazingly diverse spectrum of problems, but in the end, I think you'll see the iCons students send one clear kind of message. They send it through their courage and believing that big problems can be solved. They send it by valuing a diversity of different kind of perspectives, and in today's world, we really need that. In the end, I think they send a message of hope. And if there ever were a time when we as a society, we as a country need a shot of hope, now is the time. So it's time to hear actually from our iCons class of 2021, the so-called 8th color of the iCons program. So it's time for me to shut up and for the students to actually start their presentations. And at this time, what we're going to do is, just to give you a sense of how this will work, we're going to have one keynote presentation. Then we're going to split up into 3 parallel sessions. We'll explain all that later. And then we're going to come back and have 2 more keynote presentations. And then we're going to have the graduation event. So that's how the rest of the 3:30 to 5:30 segment will go. And so to begin, we're going to stay together to hear the very first keynote talk This is given by Joshua McGee. He's graduating with a major in chemical engineering. He's a very decorated student, having won a Rising Researcher Award from UMass, as well as an NSF graduate fellowship. And I asked Joshua's research advisor, chemical engineering professor Sarah Perry, to give the official introduction to Joshua. Professor Perry?
Hey, thanks, Scott, for the opportunity to introduce Josh. Yeah, I don't want to say too much, but Josh really embodies a lot of the sort of strengths that Scott mentioned in terms of being able to tackle problems. And the work that he's going to talk about today, he's been working on since 2018 when he joined my lab. And it was really a project that he identified and brought to the fore and then took over full ownership of. So with that, I'm going to turn it over to Josh and let him talk about his research on microfluidics for nanoparticle synthesis, purification, and characterization. Josh?
Thank you, Professor Perry, for that wonderful introduction. And hello, everyone. I'm Josh McGee, and as Professor Perry mentioned, I'll be talking about microfluidics and how flow can be utilized to improve nanoparticles. And the motivation behind this work is to introduce targeted drug delivery. And the idea here is that as we improve the delivery of therapeutics, we can improve their safety and efficacy. And so to illustrate this with an example, here is a patient that has lung cancer. And how these patients are often treated are through intravenous chemotherapy. And this type of treatment approach is untargeted, meaning that the drug is often distributed throughout their entire body and can have many serious side effects that can damage healthy tissue. A much more advantageous approach would be to have the treatment be targeted directly to the site of diseased tissue in order to protect the healthy tissue, but also to potentially improve efficacy by concentrating the drug to where it is needed the most. And so we're interested in developing technologies that enable this type of drug delivery strategy. And a particular technology that seeks to do this is called nanoparticles. And in the context of the current COVID-19 crisis, nanoparticles have saved us with the introduction of vaccines. And the way that this works is that you have mRNA that encodes for a spike protein present on the virus encapsulated within a lipid nanoparticle. And these nanoparticles can be injected into a patient. They're then uptaken by cells where they're able to present a protein that creates this cascade of immune responses, with the end result being that you're protected from COVID-19. And this is great. However, there are some challenges associated with this current technology. The vaccines require a tremendous amount of cold storage, and as a result, they're expensive, and there's some logistical constraints associated with the vaccine. And so this represents an enormous opportunity. And this is one of the reasons, on top of many others, that I'm interested in protein nanoparticles. So protein nanoparticles, like many other materials, are highly stable, but they're also inexpensive, biocompatible, and they can be used to deliver vaccines and drugs. And so the scope of my research covers three main topics, and they all focus around the use of microfluidics and flow to improve the clinical translation of protein nanoparticles. And so I'm interested in utilizing microfluidics to create nanoparticles and tune their properties, but also to purify them, remove toxic chemicals, and then lastly, to analyze their performance. So to look at their interactions with their targets, like tumors, for instance, in order to enable the possibility of replacing animal models. And so for this presentation, I'm focusing on this first aspect, protein nanoparticle synthesis, and how to tune their properties by taking advantage of flow. And so the properties of nanoparticles will affect their performance drastically. And for drug delivery, there's three main characteristics that we care about: dispersity, size, and charge. And for drug delivery, we want monodispersed particles, which means that they're very uniform, they have similar properties, and their performance can be predicted precisely. And we also want to have control of their size and charge, because these two factors will influence the different types of tissues that they're able to be delivered into. And all of these properties are controlled during the synthesis process, where controlling mixing is really the most critical factor that will determine these resulting characteristics. And with the traditional synthesis processes available, it's difficult to control mixing. As a result, it's difficult to control the resulting nanoparticle properties. And so my solution to this that I propose is to use microfluidic flow to precisely control mixing and to control the resulting properties. And so basically I'm taking this process, scaling it down to a small scale where I have really precise control of the solution conditions to better synthesize protein nanoparticles. And so for my thesis, I've created a platform to study microfluidic protein nanoparticle synthesis. And the way that this works is you have two inlets, one corresponding to a solvent, one corresponding to a protein. And so these two liquids come in, they mix, and nanoparticles are able to be formed throughout the length of this microfluidic device. And so I'm interested in varying some microfluidic parameters like flow rate, concentration, pH, and the type of solvent utilized, and determining their effect on resulting physical properties like size, polydispersity, as I mentioned, stability, and surface charge. And one of the key results that I've been able to discover thus far is that nanoparticle size can be manipulated by changing the fluid flow rate throughout this microfluidic chip. And this is important because, as I mentioned, the size of the nanoparticles will dictate the type of applications that they can be utilized in. And so the experiment that I did was I varied the flow rate through the microfluidic chip and measured the result of the effect on the nanoparticle size. And what you can see here is that there's this trend of decreasing size with increasing flow rate. And one hypothesis that I have about what is going on here is that at these slow flow rates, what's happening are that there's less nanoparticles that are forming initially. And so because there's a finite amount of protein available, the nanoparticles are able to grow more throughout the length of the device. In contrast, at higher flow rates, we have more nanoparticles forming initially, but due to the decreased amount of time that they're spending in the device, they're able to grow less. And so this is a really important discovery because, as I mentioned, small nanoparticles can open up new applications. So for instance, if we can make nanoparticles below the 100 nanometer threshold, maybe we can deliver drugs across the blood-brain barrier and open up a whole series of applications in treating some serious diseases. And so, in conclusion, I have discovered throughout my thesis work that nanoparticle properties will drastically influence their performance, and that protein nanoparticles are ideal drug carriers for many different reasons. And that lastly, we can use microfluidics to control nanoparticle properties and potentially open up new drug delivery applications. And here is a list of some future works that I have in mind, and a lot of these are covered in the rest of my thesis work. And now I would just like to take a moment and thank some individuals that have contributed greatly to the success of my honors thesis. So Professor Sarah Perry, Professor John Clear, the members of the Perry Lab, but then also the wonderful students in iCons and the wonderful faculty, and then the Commonwealth Honors College at UMass Amherst. And I would like to open it up to any questions from the audience. Thank you.
Okay, thank you so much, Josh. I would actually, at this point, I would like to ask us all to come off mute. See, if we were actually in a real place, we would clap loudly. And Josh really deserves that, and all of our students do. So I'm going to ask everybody to come off mute and clap really loudly for Josh so he can hear it. Thank you. Thank you so much. You can go right back onto mute now. So the other thing you should know is that Josh woke up this morning. He told me this. He woke up this morning excited to answer your questions. Please, 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 we have lots of time budgeted to answer your questions. And we welcome questions from everybody, from students, from faculty, from staff, from guests, from parents, whoever. Fire away, please.
Scott, can I ask a question?
Yes, please.
So looking at the differences between the protein nanoparticles and the lipid nanoparticles, how challenging is it to have other protein expressed on the surface of protein nanoparticles? So if I'm thinking about the nanoparticles, I thought you could also have things expressed on the surface so that potentially you could target those nanoparticles. Can you do the same thing for protein particles?
That's a great question. And that's actually one of the many reasons why proteins are ideal nanoparticle materials, because of the fact that they have many different amino acids that can be selectively targeted in different chemistry approaches. So you can actually functionalize the surface of protein nanoparticles with many different ligands like antibodies, nucleic acids, fluorescent labels. So the amino acids are very amenable to chemical modification. So as a result, protein nanoparticles are great because they're compatible with a series of hydrophobic, hydrophilic drugs, but then also they can be modified through the introduction of, as you mentioned, other proteins to target them to specific tissues.
Wow, oh, that's great. Thank you. Learned something new.
All right, thank you so much. We have time for more questions. You can hear my cat in the background. Yes, please, Emily.
I'm wondering what your next steps look like with this, because it sounds like you have some really cool ideas, and I'm curious what that looks like for you kind of off the end of this presentation.
Yeah, so I didn't have time to touch too much on these other two aspects of my work, but really the overarching goal that I have is to combine these three different avenues of research. So I talked about this aspect quite a bit, the use of microfluidics to synthesize nanoparticles, but there's also a great deal of other applications that microfluidics can enable. So one of the main challenges actually with the synthesis of nanoparticles is making sure that they're ready for use in the clinic. So making sure that they're safe and that all of the excess chemicals used in the synthesis process have been removed. And so there's actually a great deal of work that's being done, and I've done a little bit of work myself in my thesis to study nanoparticle purification. So taking advantage of flow to be able to remove out these potentially dangerous chemicals. But then there's also this other really interesting field right now, utilizing microfluidics to recreate physiologically relevant environments. So for instance, making things like an organ on a chip or a tumor on a chip that effectively recreates a biological environment without the use of animal models, for instance. So being able to study how drugs and nanoparticles can interact with their targets, but without actually needing the animals and the life aspect associated with those types of studies. And so really my goal would be to create a scheme that is able to combine all three of these technologies together. So you synthesize, you create nanoparticles, they're then able to be purified, and then you have this way of measuring their performance in a high throughput manner to basically be able to determine the next high functional therapy for many different diseases.
Fascinating.
So cool.
Thank you. Thank you. Thank you so much. Let's see. I think according to the grand schedule, we actually have 1 more minute. So we have time for 1 more question, if anybody would like to ask.
Hi, Josh. I have a quick question. Great presentation. Can you just describe any other results you found, maybe about how microfluidics and like flow rate affect the kind of charge ratio at all? I know you don't have a graph for it, but if you just speak on that, that would be great.
Yeah, so thanks, Dalton. That's actually a particular property that I haven't gathered too much data on, but another result that I can describe that's fairly interesting. So I showed this result here, that the overall flow rate has this effect on nanoparticle size. But another parameter that I investigated is concentration. So basically the ratio between these two flow rates. So the way that this works is you're mixing solvent with protein. So for instance, what happens when you have more solvent or more protein? And I've been able to identify some pretty interesting trends there as well, that it's not exactly a trend that you would expect to observe. And so I've been able to find that the amount of solvent and the amount of protein can drastically influence nanoparticle size as well.
Awesome. Great work.
Thank you.
Wonderful. I think everybody, can I ask you, so can we do this? Can we always, when we thank the speaker, let's always come off of mute, make lots of noise, and then go back onto mute. So let's all please give Josh a big round of applause. Fantastic. All right, thank you so much. So now we pivot to the parallel session part. And I have some notes here that I have to peek at. Yes, so we're breaking up into 3 parallel sessions. So we're going into Zoom breakout rooms. And let's see, Wesley is, let's see, yes. So Wesley is putting a link into the chat. And if you hit that link, that will show you the program. I can also show you the program. So let me bring that up real quick. Here's the program. Here's sharing the screen. Yes, that's what I want to share. Always be careful to share the thing you want to share. And so here's the program. We have 3 impressive parallel sessions. Wesley has put the link to this into the chat. So just take a moment to take a peek at this and see where you'd like to start. Session 1 is moderated by instructor, iCons 1 instructor, Dr. Christiane Healy. Session 2, moderated by iCons 2 instructor, Dr. Ali Hunter. Session 3 is moderated by iCons 3 instructor, Erica Light. Now, these different sessions don't have any particular subject focus. Each one covers sort of the diversity of the different kinds of projects. So just take a peek at this and see where you might like to start. The other thing is that you have to realize is that you can actually move from one breakout room to the next. And now I also have to say that if you have an older Zoom program on your computer, you may not have that ability, and you may have to say, hello, my name is whatever, please put me into breakout room 1 or 2 or 3, and then Wesley will send you where you would like to go. Now at this moment, that is my prompt to ask everybody to recognize the program manager of the iCons program, Wesley Dunham. There's always somebody behind the scenes who does a ton of hard work to make something like this happen. So I'd like to ask us to come off mute and make a lot of noise to say thank you to Wesley Dunham. Okay, so I'm going to stop sharing my screen. Wesley, you've made the breakout rooms, is that true?
Yes, I can open them whenever you want.
Okay, so please open up the rooms, head to the parallel sessions, and we're going to come back from the parallel sessions. So that's going to take us from now till right up before about 4. No, that can't be right. It's the transition to parallel sessions. Yes, the parallel sessions take us to about 4:50. So from now till about 4:50, please head to the parallel sessions and enjoy the amazing science, technology, engineering, and math that you will see. Thank you. Well, everybody, I'm so delighted to have you all back. That was, amazing to hear all about all of those projects. The only thing that killed me was I wanted to be in 3 places at once. And that was the only problem. So I'd like to right now just please ask everybody to come off mute, join me in giving all those students a terrific round of applause. Yeah. Fantastic. Thank you. Okay, so now we pivot and we have 2 more keynote talks. The next keynote talk will be given by Josiane Rosario, an environmental science major. And to introduce Josie, we have geoscience professor Ray Bradley here to give the introduction. Professor Bradley?
Thanks very much, Scott. It's certainly my pleasure to introduce Josiane Rosario, who's been carrying out a research project on sediments that were recovered from the bottom of a lake in the Norwegian Arctic, not far from where this picture was taken behind me. The Arctic's been warming more rapidly than other parts of the world, and so we wanted to understand how unusual the recent warming has been. And so by studying the sediments, which have accumulated over time in the lake, we get a window on the past. So Josiane's been doing this work. She's a senior from Brockton, Mass., the first in her family to attend and graduate from university. She decided to major in environmental science while she was at high school, building on an interest in earth sciences, biology, and chemistry. And because of her general interest in the environment and the environmental changes that we all see around us. To undertake the research that you'll hear about in a minute, Josiane had to overcome quite a steep learning curve when she started. But thanks to the excellent support and guidance of Dr. Francois Lapointe, who's a postdoc in my research group, Josiane stepped up and embraced this opportunity for lab work, data analysis, and a little programming as well. And as a result, she's made great progress and produced some fascinating new results, which she's going to tell us about. I just want to conclude quickly by thanking Francois Lapointe very much for his role in this project. He was front and center in the lab with Josiane over the past difficult year. And on behalf of Francois and myself, both of us are also first generation college attendees like Josiane. I want to say that we're proud of your efforts, Josiane, and I wish you all the best in your future career. Hope one day you'll be able to visit the Arctic like you here in Svalbard and see for yourself. It's a lot more interesting than staring at those sediments in the lab. So over to you.
Thank you, Ray Bradley, for that introduction. Share my screen. Okay. So, hi, my name is Josiane Rosario, and I will be telling you about my research about analyzing the physical properties of lake sediments and Svalbard. And I hope that one thing you get from this presentation is that the earth below us holds so much information about its own past. And so the lake is located in Svalbard, which is a territory of Norway. This is Norway right here, and this is Svalbard over here. And the lake is located in the southwestern region of Svalbard, and it's called Lake Linnevatnet. And this is a proglacier lake, meaning that there is a glacier called the Linnebreen Glacier nearby. And so, as Ray mentioned, Svalbard is one of the most regions that are heavily impacted by global warming. And however, the data on past climate in Svalbard only reaches to about like 100 years. And so it is important to look at the climate in a longer period to see if this recent warming in the current decades is unprecedented or not. And so what we do know is that the Linnebreen Glacier is melting away, as we see by aerial photographs in this photograph, the extent of the glacier in 1936 and the extent of the glacier in 2019. However, we do not know whether this is unprecedented in the context of past climate. So that is why in my research, I looked at lake sediments to track glacial activity over the past 2,000 years. And this is because the Linnebreen Glacier actually sits on coal-bearing sandstones. And so we can analyze organic matter content to determine glacial activity. For example, in this very simplified diagram, if glacier size was to increase towards the lake, it would be eroding coal particles and depositing them into the lake, which means there would be higher organic matter content, meaning the glacier size increasing, meaning that the temperature during this time is colder. And then the opposite of that would be the glacier retreating and therefore having no impact on the lake and low organic matter content. And so the core that I looked at was taken at Site C, shown in this photograph in the left. And the core is about 5 meters long, split into sections, each 1 meter. The picture on the bottom is in one of the cores that I sampled that is 1 meter long. I only sampled 3 meters of the 5 meter core, and the whole 3 meter core, I took around more than 300 samples. And to measure organic matter content, I use loss on ignition, which you basically put the samples in a furnace and at 550 degrees Celsius and burn off the organic matter content. And so this graph summarizes the main results from my research. And we see that between 300 centimeters to around 70 centimeters, there's a normal trend in climate or normal fluctuation in climate. And this correlates to the time period between 500 AD to around the 1500s. However, highlighted in blue, we see a big drop or increase in organic matter content. And this correlates with the Little Ice Age. And this seems to have been the coolest period since 500 AD. And then we see another dramatic trend, which is an increase in low organic matter content. And this period correlates with the past couple of decades, and it shows that there is a rapid increase in climate temperatures and also decrease in glacial activity. And this also is the warmest period seen since at least 500 AD. And so what we observe is that further and clear evidence has shown that climate change is beyond the normal fluctuations, and current increase in climate temperature is unprecedented and must be slowed down, as it has a huge impact on our environment, such as the decrease of the Linnebreen Glacier. That is all I have for my presentation. Thank you for listening. Any questions?
Thank you so much, Josiane. So we definitely have time for questions, and we know all the speakers are hungry to hear your questions. Josiane is no exception, so please come off mute, fire away.
Josiane, what type of analytical tools did you use to analyze this data?
I used RStudio to create the graphs that analyzed the data, which can be a pain, but you end up learning to love it.
A question on the global warming concern. The temperature is definitely increased dramatically, as you show, as compared to a standard. I wonder if God decreed back in the year 500 AD that was the ideal temperature, and anything that's different than that is something we've got to control at whatever cost. I'm speaking the question of what are called the warmer deniers. And they would like to say that the global warmers are the non-denier deniers. So anyway, is it your thesis is it's critical to get back to somewhere which was a norm for quite a long period, because all was in balance and the world was going on beautifully, I guess.
Yeah, so between 500 AD to 1500s, there wasn't really any dramatic changes, as you can see in the graph, and it shows that the only two dramatic changes we've seen since 500 AD is the Little Ice Age, which was the coldest time period and also the recent warming period.
Well, suppose your chart went back another 500 years, which you can't do, I guess, and you found that the temperature was higher than today. What would that say? We ought to get back to the ice age, or what should it say? I'm not giving personal beliefs. I'm giving you what you're going to face when you go out in your career, because that controversy rages on. Did God say that 500 AD was perfect? Let's get back there at any cost, or it was just happened to be the temperature at that time, and it might have been 500 degrees, not 500, but a lot warmer, not too much before that.
Yeah, I think it depends on what was going around that time. There would need to be more analysts going back further to look at the higher temperatures and trying to look at what was also going on and what could have contributed to it. But with my research, we see that that this increase in climate temperature is correlated with the start of the Industrial Revolution, because it does start to increase around the 1800s. So,
but then it went down between 45 and 53, and
yeah,
that's been attributed to the presence of global cooling elements, and that's a controversial point on its own, I think.
Mm-hmm.
In other words, the CO2 is going up and the temperature is going down, and it's explained by the fact that the elements that were going out in the atmosphere were actually cooling things.
I had a question. I'm just curious about what inspired your research.
I think just looking at past climate change through looking at sediment is really interesting. So I really wanted to take part in this research, and I really wanted to use this opportunity to see what climate was like in the past. And it was really cool looking at it.
All right. Well, I would like to thank you so much, Josie. I'd like everybody to please come off of mute and give a rousing round of applause.
Josie, that's a nice piece of work you're doing.
Thank you.
Okay. Congratulations, Josie. That was really fantastic. All right, so we're going to pivot. We have our third and final keynote. This will be given by Miranda Boudreau, a psychology major, and she will be introduced by her research advisor, Professor Kirby Dieter-Deckard of the Psychological and Brain Sciences Department. I'm delighted to ask Professor Dieter-Deckard to give the introduction.
Thank you. So I'm Kirby Dieter-Deckard. I'm a professor in Psych and Brain Sciences, and it's my pleasure to introduce Miranda Boudreau. Miranda is not only an outstanding scholar, but a dedicated emergency medical technician, or EMT, in Springfield and at UMass. Miranda joined our lab nearly 3 years ago, Miranda. That's a long time. And it's been an instrumental member of our team in examining the development of and changes in individual variations in kids and teenagers' behavioral and mental health outcomes. What I admire most about Miranda, your work, is your dedication to applying rigorous scientific and statistical methods to answer questions that have real-world implications for health and well-being. Miranda, you're receiving our department's Outstanding Thesis Award next week. Congratulations for that. It's very well deserved. And I know I speak for everyone in saying we're looking forward to hearing your research presentation today. Thank you. Take it away.
Thank you so much. All right. Well, hello everyone. Before I start, I'll kindly ask that you type your questions in the chat as you think of them. That way I can answer them at the end of my presentation, and maybe that'll make it easier for you to come up with things to ask at the end. So today I am excited to talk about how my work attempts to address two societally significant questions related to health outcomes. The first is, how can substance abuse and unplanned pregnancy rates be decreased? And this is important because 19.7 million Americans are battling a substance use disorder, in addition to 45% of American pregnancies being unplanned each year, which is nearly 9 times that of other developed countries. These shocking statistics beg the question, how can substance use and unsafe sex be identified earlier, such as during adolescence, when these behaviors are likely to begin? And to clarify what I'm referring to, as the name suggests, health risk behaviors are behaviors that impact an individual's health. The very nature of these behaviors are preventable, and therefore identifying where and how to minimize their engagement also means mitigating their linked outcomes like substance abuse and unplanned pregnancy. In addition, individuals with depression and anxiety have been shown to disproportionately engage in these behaviors. This is particularly significant for adolescents because as many as 46% experience clinical levels of anxiety and depression. In addition to only a portion of these individuals being diagnosed, even more that are undiagnosed just don't present with high enough levels of symptoms. Next, internalizing problems is a broad term that describes the hidden issues related to one's internal or mental state, and therefore can be a measure of depression and anxiety. When measured using a questionnaire, it can inform about 3 different scales that make up internalizing problems. The first is anxious depressed, which can be feelings of being tired or not enjoying activities. Social withdrawal, which is wanting to be isolated from others. And lastly, somatic complaints or physiological symptoms like headaches and stomachache. And this literature informed my question, does engagement in health risk behaviors differ for adolescents with depressive and anxious symptoms? And so first, I hypothesized that internalizing problems, which was a measure of depression and anxiety in adolescents, would be positively correlated with health risk behavior engagement. Next, I wanted to determine if certain subscales of internalizing problems would serve as a better predictor of engagement. And for this, I hypothesized that high scores of these subscales, in particular somatic complaints, which have been shown to develop as a result of depression and anxiety, would be positively correlated, again, with health risk behaviors. And these hypotheses were answered using data from a previously collected longitudinal study out of Virginia Tech, where they recruited 167 adolescent and caregiver pairs. And my study used the final wave of data where the average age of adolescents was 17. To test my hypotheses, internalizing problems were collected using a modified version of Youth Self-Report. This questionnaire had adolescents report on their emotional and psychological state of somatic complaints, anxious, depressed, and social withdrawal, the previous subscales I had mentioned. Next, to determine engagement and health risk behaviors, data was collected using an abbreviated Youth Risk Behavior Survey. And this measure was only reported by the child and asked questions related to their substance use of alcohol, marijuana, and cigarettes, as well as risky sexual activity. And so together, these measures were used to determine adolescent engagement and were controlled for by these variables. So using the scores from the questionnaire data, I ran a test called the bivariate Pearson correlation, where internalizing problems or one of the subscales was present on the x-axis, and health risk behaviors or one of the other subscales were on the y. And since I hypothesized that there would be a positive correlation between these two variables, a significant correlation would look something like one of these trends in these two graphs compared to this one that shows no association between the two variables. And surprisingly, I found there to be statistically significant correlations between somatic complaints and health risk behavior for nearly every group I controlled for, but not for internalizing problems, anxious, depressed, and social withdrawal. And these results may suggest that health risk behavior correlation with somatic complaints may be unlinked from internalizing problems, and therefore may be unable to indirectly link anxiety and depression with health risk behaviors. Despite this, these results indicate that somatic complaints provide a robust measure of detecting at-risk adolescents. Although I was not able to determine whether somatic complaints cause engagement in health risk behaviors, they inform us that they could potentially be used to screen adolescents, as well as what preventions or interventions might be most appropriate. This is especially relevant as this relationship cuts across gender and most income to need ratio. Thank you so much for listening, and a very big thank you to the iCons program, Kirby Dieter-Deckard, and Young Min Kim Spoon for making this research possible. And I'm happy to take any of your questions now.
All right, thank you. Everybody, please join me in a round of applause for that amazing talk. Okay, and we have time for questions. We actually have some in the chat. So Miranda, do you want to, would you like me to field the chat or?
Yeah, that would probably be easier.
Okay, so first question is actually about the software used to make the talk. What software did you use to make the presentation?
This is PowerPoint. I had downloaded a really cool design from Slides Go or something like that, and I just kind of modified it to my presentation. So
got it. Okay,
not
cool.
Unfortunately, not my creativity, but
good old-fashioned PowerPoint. Okay,
yeah,
great.
exactly.
So a second question: how long in time? So period of time was this study conducted over?
So it was 4 years. I can go back to that first one. So basically it was 4 different waves, and every year the same group of adolescents were surveyed again for each of these things that were looked at. But for my study, I only used the 4th wave, just because during this time, it's usually when adolescents are more developed, maybe more likely to have already engaged in these behaviors. So since I was only looking at one group at this time, it seemed like it would be the most informative. But I did consider doing this longitudinally, looking at between wave 3 and wave 4 and how trends kind of would change, whether they would look different for different ages. But just for the sake of time, I only looked at the 4th wave.
Got it. All right, so we have 3 more questions. Keep on coming. Do health screenings currently inquire about somatic complaints?
As far as I'm aware, the ones that are given in pediatricians' offices don't ask questions related to this specifically. The questionnaires that are generally asked get at more of the anxious, depressed, and social withdrawal scales. And so maybe this is just another thing that could be tacked on to either that questionnaire in pediatricians' offices or something entirely different.
Okay, real world impact of your work. All right, we have time for one more, and it is, would you expect to see different findings with data sourced from different regions in the country?
That's a really good question. And based on the diversity of my sample, obviously you can see that very not diverse ethnically, but in terms of the region that this was collected from, it was representative of that area. I would maybe expect some differences, but when I looked at becomes the need ratio, which is kind of a similar measure of SES. I did not find I did find significant correlations between somatic complaints and these things. However, my sample was pretty small when I separated them out, so I might not have had enough statistical power to find significant results for this in particular. So potentially, but kind of would need to look a little bit more.
Got it. All right. Well, Miranda, congratulations. Everybody, please join me in a big round of applause for Miranda.
Thank you all so much.
Miranda Boudreau. All right. So here we are. We are actually, we've completed the student presentations, and I don't know about you, but I have two words: wow and wow. That was excellent. I think you all agree that UMass Amherst is blessed with some amazing students. So let's, for all the students, all the keynote presentations, Joshua, Josie, and Miranda, and all of the talks that we heard today, one more big round of applause.
Scott, Dick Mahoney,
Yes.
most of the presenters gave a great deal of credit to the iCons program, and since you are Mr. Icons, I think you ought to get a round of applause as well.
That's very kind of you. Very kind of you. You can tell that to my wife. Just kidding. Okay, so here we are. We are at a very important stage, iCons seniors. You've taken all the iCons courses, 1, 2, 3, and 4, creatively named for the 4 years of college. You've tackled real-world problems throughout college. You've engaged repeatedly in this icon cycle of inception, engagement, research, create, and reflect. And now you've completed your final presentations. And so by the power invested in me by the University of Massachusetts Amherst, I am proud to say you have completed the iCons program. You are our 8th cohort of iCons alumni. Congratulations. Awesome. And at this stage, I would like to recognize an iCons alumnus, Eric Wusthoff, who is a leading member of the iCons Alumni Network, and he is going to come forward, and I think Wesley is going to make that happen using his special powers, and Eric is going to welcome you into the family of iCons alumni. Eric, take it away.
Thank you so much, Scott, and congratulations to everyone. I am iCons 6th cohort, class 2019, and so it's not too far away from being where you all are. I remember being relieved and a little sad, but mostly relieved at finishing my senior presentation. So congratulations to you all. Scott let me know earlier today that officially, as of minutes ago, the iCons alumni network is now larger than the iCons program because there's about 200 students in the iCons program, and now they have lost a good chunk of you, and you are now on the dark side to the alumni. So I want to welcome you all. The iCons Alumni Network is an amazing group of people. You all know how amazing iCons people are because you've spent 4 years with each other. And just imagine a bunch of them, but now they're all actual adults. And so we do a lot of work around supporting iCons students through events. When it's non-COVID times, we also get together in the Boston area, but increasingly, we've been trying to pull in the network from not just Mass, but from all across the country and the world, because as I said, it's the iCons alumni are only getting bigger and bigger and spreading across the world. And so I'm going to pop the email for the alumni network. If you have any interest in getting involved, we're really excited to continue to serve our students, and we encourage you to be involved. We have monthly happy hours where we kind of just hang and chat and give updates about our lives. And then we also have programming for students in the iCons program. And we really welcome ideas. We're a really scrappy group of people who care a lot about the mission of iCons and taking that mission into our future careers and graduate schools and med schools and everywhere in between. So we really welcome you. We hope to see you. We know many of you have already been involved in our events or been involved with our programming. So feel free to reach out with any questions. I'll pop my email in there too, but the team email is there for you to reach out, and please get involved because we're so lucky to have 36 more of you tonight.
Thank you so much, Eric. Awesome. All right, so we have one more really big task. This is going to be like a little icons case study in and of itself. Is that what we have to do right now is we have to take that final icons picture. You all remember that icons picture that we took when you first joined the program, that fateful December Saturday. What are they asking me to do on a Saturday morning? We got together, we did that process where you picked your icons 1 case studies. We took that picture, that was then, and this is now. And so in order to do this, there's a few steps. Step 1 is I ask all seniors to turn their video on, and I ask everybody else to turn their video off. And Wesley, I ask you to unpin me. So, so I'm going to turn my video off right now. And so that's the first step. The second step is that all seniors received an envelope in the mail, and that envelope contains 2 items. Oh, I should say, have that envelope in your hand, but don't open it yet. So that envelope contains 2 items, a certificate and a cord. Now there's a certain mythology about the cord. The idea is that each individual strand on its own is kind of weak, but taken together, woven in just the right way, it becomes very strong. It can't be broken, it can't be torn. So just like each individual field is not able to solve these really complicated problems, but through collaboration and interdisciplinary work, we become very strong. So that's the mythology behind the cord. So what I ask everybody to do is when they open up their envelope, please wrap their cord around their neck, but do it safely. We don't want anybody, choking. So wrap their cord around their neck so we can see it, sort of like a necklace. Please hold up the certificate so we can see the certificate in your Zoom screen plus your smiling face. And we're going to take a bunch of screenshots, and keep on smiling because it takes a little while to take all the screenshots. So it could be that we're seeing that some people don't have their envelope. And if that's the case, that's fine. We will, okay, some folks, I don't. Okay, well, regardless of if you have your envelope or not, don't worry about it. We need to see your smiling faces. So if you have the envelope, please feel free to open it. If you don't, no problem. We just want to see your smiling faces. Please open it up, take out the certificate, hold it up, smile for the camera. Wesley, please
Yes.
do what you need to do to take the screenshots.
Great. Yep. If everyone will just hold, I think it looked like 3 photos I got to do.
Mm-hmm.
So keep holding. And I still see people moving, so I'm going to wait for a minute.
Okay.
Watching Kenneth and Samantha. All right, so I'm going to start. I'll let everyone know when to stop because you don't know if you're in my screen. So yeah, keep holding.
Keep on smiling. It's a skill. Hold that smile. Keep going. This is what Britney Spears does every day. Smile, smile. Smile, smile, smile. You can do this.
All right, and all right, that's it. It was just 2 pages, so thank you.
Okay. All right. Wonderful. Excellent. All right. So so everybody can turn their video on as they like, whatever they want. And at this point, we are just at the end of the Senior Expo. We now are going to quickly pivot to the Senior Celebration. But before we do that, we have to give thanks. An event as big and momentous as this can't happen from just one person alone. It always takes a village. There's definitely a team of iCons faculty and staff, and now it's time to say thank you. So to all the faculty who've taken the scary leap of teaching in this weird icon style, walking into a classroom and actually asking students, what is it that you'd like to learn? What is it that you'd like to work on? As opposed to the normal way of walking in with the predetermined syllabus, I want to say thank you. To all the staff who cared for and advised each and every student, helping them to navigate through the complicated waters of both the iCons program and their major, I want to say thank you. To the administrators who took the risk of supporting a program with no precedent at all. We didn't know how to teach and we didn't know how to pay for it when we launched the program, but our administrators knew this was still an important thing to do. I want to say thank you. To the parents who are with us right now, who sent their children to the Commonwealth flagship, the big school, hoping that they would find their people, I want to say thank you. To our many friends and sponsors without whom we wouldn't be here today, I want to say thank you. But most of all, to the students who dared to apply to the program, join it, and complete this rigorous training in this year, the year of COVID all year long. You made it to the end. From the bottom of my heart, thank you for making this journey with us to this day and making it together. Congratulations. And this completes the 8th annual iCons Senior Expo. We now pivot, because we pivot on a dime here, we pivot to the next part of the show. And that means I have to scroll down my page. Give me a second to scroll. So the next part is it is 5:30 on the dot. This is Wesley Dunham, so organized. Thank you, Wesley, for keeping us right on time. We are at the Senior Celebration. So normally, in a normal year, we'd be leaving a room like a academic building, classic kind of lecture hall, and we move into a more celebratory space. There'd be beer and wine flowing, maybe some music playing. We'd be having fun, which we are right now, of course. We are having fun. So I promise to you, I'm not going to sing to you right now. So that's my gift to you. But I do invite you to enjoy whatever beverage of your choice you would like. Now, there could be some people who are just joining us now. And if you are, it's my pleasure to welcome you to the 8th annual Senior Celebration. I'm still the same person. And that's, for some reason, I get stuck on my name sometimes. Somebody blurted out.
Scott Auerbach.
Scott Auerbach. Thank you very much. Scott Auerbach, the Mahoney family sponsored executive director of the iCons program. And that right there is my cue to talk about the Mahoney family, to gratefully acknowledge the generous support from the Mahoney family. The three brothers, Dick, Bob, and Bill, have been incredibly supportive and committed alums of UMass, all coming from chemistry. And I understand that we have Bob Mahoney with us right now, and I'm going to give just a quick intro to Bob. He's going to make some remarks. So Bob Mahoney graduated with a chemistry degree from UMass in 1970. Bob's been a staunch supporter of iCons, like his brother Dick. Bob is also a sort of a master, like a Jedi. He's in the Jedi Council, as it were, of career counseling. He works with our students, and he does something that's incredibly difficult. He helps them to figure out what's the difference between a job and a hobby. He was most recently president of Belmont Savings Bank, and although Bob didn't follow his brothers into the field of chemistry, he often says that his rigorous training has given him the intellectual confidence to have the success that he's had. So Bob, thank you so much for coming today, and the floor is yours.
Thank you, Scott, and congratulations to you and your team, and most importantly, congratulations to all the seniors. I got a chance to listen to some of the projects, and I have to admit, notwithstanding my degree in chemistry, I understood the words, but the sentences defied me, and a few of the concepts, most of the concepts defied me. But it was great to hear these brilliant young people talk, and to be honest, I envy them. I really envy them. It was 50 years ago that I was in their shoes, getting ready for the next step. And as to quote my brother's favorite statesman, Winston Churchill, he reminds us that this is not the end. This is not even the beginning of the end. This is just the end of the beginning. And so as these brilliant young people go off into the world, they have ended the beginning. And I encourage all of you to use the skills that you've learned at iCons to enhance your career and have really a wonderful, wonderful career, to really do something you love. Because if you find something you love, you'll never work a day in your life. And iCons was designed 13 years ago to give people a multidimensional, multilateral view of the world, the way Mother Nature actually thinks about life and thinks about the world. It's not physics in one line and biochemistry another line. It's all integrated. And so as you think about the way you've learned to think about things in multidimensional ways. I hope you think about your careers that way. Most of you don't have a clue what you want to do, and that's perfectly fine. I'd be very suspicious if you're convinced you know what you want to do. If you're convinced you know what you want to do, think again. It's too soon. But use the skills that you've acquired to do research, to inquire, to ask the questions, to experiment, and to try things that are a little outside your comfort zone in that goal to find something you love, because it is nothing like it. I graduated in chemistry and then promptly went on to spend 48 years in banking. Go figure. It turns out I didn't like chemistry that much, but I sure as hell love banking. And I just had a wonderful time doing it. So it's a very exciting time in your life. I truly envy you, and the opportunity to start a new life with all you've learned. And I just encourage you, don't give up. Don't give up until you found something you love, because you deserve it. You deserve to do something you love 7 days a week for the rest of your life. Good luck.
Thank you so much, Bob. Thank you for those wise words. I remember learning from my dad, watching him. He loved what he did, and it inspired me. It gave me the idea it's possible to love what you do. So thank you, Bob. And thank you to your whole family, to you, your brother Dick, and your brother Bill for your support, unwavering support over these years. So now we pivot to an important part of the program, the awards part of the program. And we start with the Mahoney iCons Alumni Award. This award is in its 4th year, and I think we have at least, so that means there have been 3 previous winners, and I think we have at least 2 of them here with us today. So thank you folks for coming. This was funded, of course, with a generous donation from the Mahoney family. And this, I think, goes to one of my favorite pieces of wisdom from Dick Mahoney, which is that while process is important, outcomes are everything. And there are 2 outcomes that we look for in our alums. One is that they're out there practicing those special attitudes, knowledge, and skills they learn in iCons, and they're practicing them in the real world. And the second one is that they stay connected to the iCons program, working with our students, helping them to find jobs, and making sure that we stay true to the mission of making sure the iCons training is fresh and relevant. So the Mahoney iCons Alumni Award was designed to recognize just these outcomes. And I would like to ask Professor Justin Furman, who's the director of the academics for iCons, to come forward, describe the process of how this award was made, and of course, announce the winner. Justin?
Hi, thanks a lot Scott for bringing this to the forefront. The process for selecting the Mahoney Alumni Award is that alumni who have been out of the program for at least a couple of years are invited to submit an application and a reference letter from their supervisor. And those are read by a committee of a faculty member, some people from outside the program, and some current students to look for the characteristics that Scott described. And this year, I would like to introduce Erica Light, who was the unanimously selected recipient of the Mahoney Alumni Award this year from amongst the strongest application pool that we've ever had for this award. I've known Erica for about 10 years since she took a chance on icons back in 2012, when it was still quite experimental. Since then, Erica has been a thoughtful, vocal, enthusiastic, strong-willed, exemplary member of her icons cohort. She's been an innovator, expanding the boundaries of icons research. She was an early leader in the icons alumni network. She's been a research mentor to many icons students. She's been a partner in creating a new icons 3 biomedicine course, filling the biggest instructional need that we've had in the first decade of icons. Considering everything that she's done, to say that Erica meets the criteria of the Mahoney Alumni Award is kind of like saying that the Beatles were an okay band. The selection committee noted a few things in reviewing her application. They said that Erica applies icons principles to teaching, thinks and acts collaboratively, emphasizes metacognition and reflection, and advocates for equity in icons and beyond. They noted that Erica has taken the icons mission of shaping future generations of leaders to heart. She collaborates with coworkers in a way that is equitable and non-hierarchical, with an understanding and appreciation for the interdisciplinary collaboration that icons cultivates. And that this is just an awesome example of the quality of icons. How better to exemplify these qualities than to dedicate herself to education. So please join me in congratulating Erica.
Thank you, Justin. Thank you, Scott, for directing this great program. I want to thank the Mahoney family also for sponsoring this award. And I want to thank the entire ICONS program of staff, students, faculty, and our sponsors, without whom this program would not be possible. My name is Erica Light, as Justin introduced me. I've been at UMass for the past 9 years, first as an undergraduate in the third cohort of icons, then as a staff member, and now as a graduate student. And I feel in this moment, we're kind of coming full circle, because the first or second icons award ceremony I went to, Scott presented Justin with the icons award. And then several years later, I presented Scott with the Distinguished Teaching Award. And now Justin is presenting me with an award. So it's nice that as we come full circle also in another academic year, we can take this time to reflect on our relationships with others, what we've learned from each other, and what we want to do next. As I reflect on my relationship to the ICONS program and what it means to me, the memories that come to mind are somewhat pedestrian and mundane. I remember the first time I got in office at UMass, Justin came by to congratulate me and just sit in my office with me like I used to sit in his office when I would go to his office hours almost every week. Sometimes Scott would meet up with me for lunch. We would discuss our vision for a future I3B course. And then other days we'd meet up at the U Pub after work, and I would say, I'm just tired. I don't feel like talking. And Scott would say, you sitting here silently is enough for me. These are the kinds of things that I think really capture what my relationship to the ICONS program has been. It's been one characterized by interdependence. I've relied on the program when I've needed support, and the program has come to me when they've had a role for me to fulfill. But it hasn't just been our needs that have brought us together. We've also come together to celebrate our successes, and we've shown up for each other. When I think about my relationship to the ICONS program, what comes to mind actually is a quote from Lynn Margulis. So as many of you probably know, she's a late distinguished professor from UMass. And I'd like to share this quote from her book. My cat is also very excited about this quote. I believe that most evolutionary novelty arose and still arises directly from symbiosis. The reason that I'm sharing this quote is because this quote challenges our notion of what evolution means. It challenges the notion of competitive evolution, of dominance and hierarchy that's embedded in this logic of survival of the fittest. It argues instead that novelty emerges at the site of cooperation across difference. This is how we evolve. This is how we overcome problems in our environment. And this is how we contribute to a more complex and curious world. Throughout my time in the iCons community, my mentors have met me where I'm at. They've been willing to listen. They've been willing to exchange ideas 2 ways, and they've been willing to build trust with me. This is the approach to relationships I seek to reproduce and normalize as an instructor in the iCons program, and to tackle the challenges of our time. Thank you.
Awesome. Thank you so much, Erica. Thank you so much. Congratulations. Okay, wonderful. Congratulations. Richly deserved. All right. So now we have a special guest who's coming soon. And so this gives me a little bit of time. We're only going to have 3 minutes of Bob Langer. So we're going to make that time, we're going to use that time very well, which means I'm not going to introduce him when he's here. I have to introduce him beforehand. So the moment he gets here, we hand him the floor. So Bob Langer, a professor at MIT in chemical engineering. On some level, he needs no introduction at all, but I have a couple minutes here, so I'm going to take the time to introduce him. He is the most cited engineer in history. He is the Thomas Edison of our time. He is right now the holder of either granted or pending 1,400 patents. That's 1,400 patents. He's started 40 companies, including one called Moderna, which we probably know about. He is the inventor of a field called bioengineering, and he came to the chemistry department back in about 2000, year 2000, to give a seminar. Back then I was in charge of seminars, so I happened to be driving him around, and I asked him how he ran his lab. And he said, well, we get interested in real world problems. We assemble interdisciplinary teams around those problems. We tackle those problems and we find solutions, and then we push those solutions out of the lab. And it made so much sense to me. I thought, why doesn't everybody do this? Why don't we have an undergraduate, program for this? And lo and behold, we do. And Bob Langer is here to give some congratulations to the graduates. And I'm delighted, Bob. Thank you so much for coming. The floor is yours.
Well, thank you, Scott. And I like I said, I've been at another meeting, but I told Scott I'd be honored to step off for a minute or two just to congratulate you all. I looked at the abstracts. It's amazing. You guys are doing everything from nanotechnology to energy to biofilm. It's really been impressive for me to see the variety of things that you're all doing. And I was down there, as he said, and it's just a great program. And anyhow, delighted to congratulate you all. And I can imagine, looking at the things that you're already doing, you'll all have a wonderful future in front of you. So congratulations from me.
Thank you so much, Bob. Thank you so much. So what's one piece of advice that you can offer as these students embark on an interdisciplinary career?
Well, the advice I give, and really it's for anything. I mean, when I do a commencement speech or something like that, and it doesn't limit it to interdisciplinary career, but I think interdisciplinary career is great because it exposes you to new things. But I always like to think that what I try to tell my students is like to dream big dreams. Don't do something incremental, do something and dream about something that can change the world and make it a better place. I also say there's actually 3 parts to this. So that's the first thing. The second thing is, if you try to do something really big and really important, a lot of people will tell you you can't do it, that it's impossible. And that gets to the third part. Don't listen to them. Just follow your dreams. And by the way, you'll probably run up against lots of obstacles, and you'll have to use your judgment about whether to continue. But I wouldn't give up easily. I just think it's really worthwhile to dream big dreams that can change the world. And by the way, what Scott said about Moderna with the COVID virus, I can't tell you all the people that said it wouldn't work. Even the Boston Globe, less than a year ago, they have a headline with my picture underneath it saying, about how people don't do science this way, and just saying all these experts were criticizing what we did. And and that happens. I mean, and like I say, I've been at this for a while, but Boston Globe and places like that, and the experts they talk to, they keep telling you that trying to come up with mRNA vaccines, which are, by the way, delivered through nanoparticles based on things we did a number of years ago, they'll tell you it's impossible. And like I say, you can either listen to them and give up, or try to do something that'll make a big difference. So, to me, even though it's not pleasant when the Boston Globe and places like that try to ridicule you, you have to hang in there and try to do your best. So that would be my advice. And it's not just the newspapers, it's your colleagues and stuff like that.
Right. And funding agencies, right?
Oh, absolutely. I mean, I didn't even get into that.
I know.
They ridicule you even more.
Indeed, indeed. Well, Bob, I know that you have to run because you're in the middle of a break on a meeting, but we're absolutely delighted that you came to visit. And as you know, we've been sending you students with in need of your advice, and we're going to keep on sending them. So thank you very much.
My pleasure, and congratulations to all of you. Take care.
All right.
Bye-bye.
Thank you so much. Thank you.
Bye.
All right, so that was kind of exciting. And after we take a deep breath from that experience, we pivot to the next stage, which is the next award. The next award, or series of awards, is the Crowley Noack Award for Icon Student Leadership and Philanthropy. Now, this award is the brainchild of Dr. Peg Crowley Noack. She is a member of the ICONS Advisory Board. She's, in fact, the chairperson of the advisory board. She is president of Zypher Medical Affairs, and she has been a brilliant, generous, and tireless supporter of ICONS and its students. So, Peg, thank you so much for being here today. Would you please share with us the idea behind this award? And of course, we want to know the winners.
Well, thank you, Scott. It's great to have a chance to be here and to see everybody and to talk about the award. And first of all, it was fantastic to listen to all the science, which is really the heart and soul of the program. The communications were fantastic. The way you delivered your message, the simplicity of the way you could tell your scientific story is something that's so important. But what's near and dear to my heart, in addition to the fact that you're all getting a fantastic scientific foundation, is that you're also building an incredibly strong network of people who will be the next generation of leaders in our country, from maybe a scientific perspective, maybe from a business perspective, maybe from many different areas. But what that means to me is I think that it's really important to continue to build this network, have you together, have you working together to make this ICONS program even stronger. So I put together a scholarship to help to encourage that. So first is to identify leaders of the program. 2 leaders each year are identified to receive the scholarship, and they're leaders who are chosen by their peers in the program, chosen based on criteria that was defined by the program. I think the ICON students certainly know what they want and how to lead, and we let you handle that side of it. And once they're chosen, the expectation is that they help to encourage philanthropy across the program and across the years. And the idea is to try to put back a class gift to keep the scholarship going, to encourage and bring along a up and coming second-year student, to give them another scholarship, and to continue this cycle of giving back to the program and watching the growth of everybody who's part of this program. So it's a really exciting opportunity from my perspective to be able to say that I started that, and I'm watching it grow and hearing that there are over 200 students in iCans. I'm lucky enough to be in touch with many students who continue to reach out to me over the years, and I'm very happy to be a part of this program. So I want to just make sure I have everybody's names. So hold on one second.
I have the names too, if you'd like me to.
Just to make sure I get it exactly right, Scott, if
Yes.
you want to just announce the names, that would be perfect.
Yes, I would be delighted to. So the 2 winners, these are rising seniors. We have a natural resource conservation major named Abigail Guinan, Abby Abby Guinan, and we have a chemical engineering major named Kieran Tay. Congratulations to Abby and to Kieran.
And now I can see them. I was looking for everybody. I thought I would be able to find you easily. Congratulations, and thank you for taking on this honor. And I'm looking forward to working with you during the next year. And it'll be a great year. And also, congratulations to the class this year, because it's certainly, with work from Megan and the team, it certainly was an incredible year for fundraising. And I'm really proud to see what has happened, to see the level of giving and the contribution, not only from this class, but also from all the classes who have finished. And it's not easy to do that. And believe me, as somebody who has done a little bit of fundraising, knows some people who do this type of work, I know how hard it is. And it's such a pleasure on my side to see the effort that went into it and the giving and how much that will contribute overall to the program and hopefully to the future of UMass and iCons. Thank you.
All right. Thank
Congratulations.
you so much. Congratulations to Abby and to Kieran. I have to say I've had the pleasure. I had Abby in iCons 2 Energy last year, and I have the pleasure of having Kieran in iCons 2 Energy this year. This is one of the pleasures of not only directing the program, but teaching in the program. And whoever was in charge of picking these students did a fantastic job. Congratulations to you both. Okay, so we are nearing the end, but we're not quite there. There's one really critical step that we have to do. And as we've said many times, and you've probably heard this many times, the iCons program is a student-driven program. It's driven by the students, for the students, of the students. And so it is only fitting that in the last 5 minutes of the career of these seniors, we simply hand over the floor to them. Please realize that we have no idea what's about to come. And that's what we call trust. So I think that Aurelia Reynolds is got something in mind. So I simply hand over the floor to you, Aurelia, and the rest of your classmates in the 8th cohort. You've got 5 minutes of fame.
Thank you, Scott. I also want to invite Andrew. We have something a little prepared for you. Let me just share my screen. Just want to, in this time, really reflect on what iCons gave us and what we were able to get from iCons from each other. It's always a great time to look back at that. So here are our refractions from the prism, what we got from what you guys put in. And I'm going to give it to Andrew to say some words about our experience.
All righty, so this is it, everyone. Here we go. So, dear iCons, your impact on each and every one of us has been immense. You have made us feel like we are part of a tight-knit community at such a big school, which we will cherish forever. This program has been invaluable in helping us foster skills that will help us work with a diverse group of people on real-world interdisciplinary problems. Throughout the past 4 years, you have bolstered our leadership skills, made us comfortable with the uncomfortable, built lifelong connections, and took teamwork to another level. Our memories will outweigh the thousands of words this one picture can tell. The sheer number of memories we would like to bring to light shows us just how special our time in iCons has meant to us, but I will just name a few. The excitement from our iCons 1 presentations was the first exposure that made us all feel like we were part of the iCons community. In iCons 2, the renewable energy debate was a spectacle to say the least. And in biomedicine, we loved the entire process, but we really never thought we would be making pancakes and eating cheese on our last day of class in a college course. Overall, though, it was the little memories that made the most impact. Conversations about our futures with peers, the creative team thinking, and our reflective conversations are always going to be with us. On behalf of all of us, I would like to say thank you for believing in us and helping build up students who now have the confidence to carry out their life's aspirations. When we joined this program, we were handed the license to explore our curiosities. And even when the work seemed daunting, your support was unwavering. We are now the next leaders of science and technology, and that's all because of you. So thank you.
Thank you. We just wanted to share. We collected all of the memories, all of the good thank yous, and what we all gotten out of the program from all of the students. You know, their final remarks in one big just color mess of words to hopefully, you know, really show some language that really reflects of all of our experiences and icons for the past 4 years. So thank you to all of our peers and the faculty and staff who have made this experience so incredible. We really owe you one.
All right. Thank you so much. Thank you very, very much. That was very dear. I can't wait to read that. I always love to see that. I couldn't exactly read it in the moment, but I can't wait to read it. So thank you, really, Andrew, our 2 previous Crowley-Noack scholars. So it occurs to me that we skipped an important step. And the important step is I skipped it, not we. I gotta own it, right? I did it. And the step that I skipped is that part of the Crowley-Noack Leadership Award is that there is a scholarship that goes to a rising sophomore. And I neglected to actually name that person. So there's somebody here who is in Icons 1, finishing up Icons 1 as we speak, a rising sophomore who is receiving this award. And this person is Bellis Min, who's the winner of the Crowley-Noack Scholarship. So let's all please give a round of applause to Bellis. Congratulations.
Thank you so much.
Yes.
Congratulations.
Congratulations. And let's also please give Dr. Peg Crowley-Noack a big round of applause for her huge support of the Icons program.
Thank you.
Okay. Well, believe it or not, according to my cell phone, it's 6 o'clock. And I think that we've done everything there is to do. We've said everything there is to say. So unless there's any objection, I think we can call it. The 8th Annual Icons Senior Expo and Celebration come to a very successful conclusion. Congrats to all the seniors. Thanks to all the friends who've come. And remember, whatever happens, every real world problem is a great educational opportunity. And that's why Icons gives us hope, always. Thank you, everybody. Farewell. Be safe. Thank you for coming. Hasta la vista.
Thank you.